Thank you, Kat and Ashley, and thank you to everyone for taking time out of your evening to join us here for Under Pressure, the Neuroscience of Stress. I think every single one of us here at the Public Lecture Planning Committee is extremely excited for all of our talks tonight, both because the topic, I think we can all agree, is pretty relevant nowadays, and because we have a really, really exceptional lineup of speakers, both from Penn and, for the very first time for the Public Lecture Series, from Temple University as well. So we're very excited to get to all of our talks, and we think that you'll find them both very informative and really enjoyable. Now, before we do get to the talks, I want to give a very brief introduction, both to myself and to the general sort of flow of tonight's events. So to start things off, my name is Evan Gallagher, and I will be your MC tonight. And as the slide here says, I am a neuroscience graduate student here at Penn, which means that I'm basically stressed 100% of the time. So I feel well qualified to host an event centered around stress. I might even call myself an expert, at least at being stressed. I got that part down for sure. In any case, before we get to the talks, I'll spend a couple minutes giving a very broad overview of stress as a concept, just to sort of get us all on the same page. And then we'll be joined by Dr. Heather Hirsch, who will be taking us through a brief breathing exercise to help all of us de-stress a little bit, because I think we're all probably a little bit more familiar with stress than we might like nowadays. After the breathing exercise, we will hear from Dr. Michael Hart, Dr. Deborah Bangasser, and Dr. Allison Mackey. And then we'll wrap up the evening by bringing all of our speakers back for a panel-style question and answer session. So once again, definitely submit any questions you have using that Ask a Question button in the bottom right. And if your question doesn't get asked to an individual speaker, definitely stick around to the end because they might discuss it as part of that panel Q&A. Okay, so with all of this being said, let's talk a little bit about stress. Now, one of the things that makes stress really interesting, particularly for an event like this, is that every single one of us has an intuitive understanding of what this word means, right? We all know what it feels like to be stressed. In fact, like I said, most of us are probably a little bit more familiar with the feeling of stress than we might like to be nowadays. And that's actually one of the biggest reasons that so many scientists are interested in studying stress, right? Because it is such a universal experience, both in humans and in pretty much every other organism. You know, it stands to reason that we should want to understand exactly how stress affects our bodies and, of course, our brains. But it turns out that stress, in spite of being this very sort of familiar feeling for all of us, is actually a really complicated phenomenon that is influenced by a huge variety of factors, both within our bodies and in the environments that we occupy. And for that reason, when we're actually trying to study stress or how our bodies react to stress, we need to start being a little bit more specific about what we're actually interested in. For example, an important distinction that we can make right off the bat is the difference between a stressor and a stress response. Right? So a stressor fundamentally is anything that disrupts or threatens to disrupt our body's sort of normal way of doing things, what we would call our homeostasis or our status quo, right? By contrast, a stress response is the reaction of our body to any given stressor. It's essentially just our body trying to restore that homeostasis following some sort of a disruption. And as you might expect, the type and the duration of the stressor dramatically affects the nature and the intensity of the stress response, right? For example, some stressors might be psychological. So this could be a big exam that's coming up, or running late for an important meeting, or, you know, an increasingly severe global pandemic, or things of that nature, right? But other stressors might be physiological, right? So historically, this would be something like having to escape from a predator. Nowadays, it could be, you know, chronic sleep deprivation or traumatic brain injury or something like that. So right there, we actually have a pretty interesting question to study already, right? How do different types of stressors affect our bodies and our minds? Likewise, we can ask all sorts of interesting questions about the stress responses themselves. For example, we could ask if there's a difference between an acute stress response, right? What happens after a single stressful event versus a chronic stress response, which would be, you know, what would happen after you're stressed for a long period of time. And of course, we could also ask whether the answer to that question depends on whether the stressor is psychological or physiological or both, right? So as you can see, we're already getting kind of complicated here, but these sorts of questions are actually just the tip of the iceberg, really, because it turns out that a whole bunch of other factors can also influence our body's reaction to stress, right? And I've listed some examples here, but the big picture point I want to make with this slide is that even though we all sort of understand what stress, feels like, it turns out that actually studying stress in a controlled, methodical, and repeatable way is quite a bit more complicated than it might initially seem. So how do we go about studying stress? Well, as you might expect, there are a lot of options, and we'll hear about several of them throughout the talks tonight. But one of the first and most important decisions that anyone who wants to study stress has to make is what kind of organism they want to use for their experiments, right? On one extreme, we have single cells that we just grow in a petri dish. Now, obviously, these are pretty far removed from human beings, and that's a major disadvantage of using cells. But at the same time, cells are cheap, they grow really fast, you can produce a huge number of them in a relatively short amount of time. And super importantly, you have exquisite control over every aspect of the environment that they're living in, right? So if you're interested in how different types of cells respond to different types of stressors, this is actually a really good option. On the other side of the spectrum, of course, is human beings. And studying humans is appealing because, at the end of the day, we are most interested in how we as humans respond to stress. But as I'm sure everyone knows, human beings are really complicated. And just as importantly, they're also really inconsistent, right? Every person's life is unique, and that makes it really hard to figure out whether two people react to stress differently because their bodies are different or because their upbringings were different, right? Or some combination of the two. And that's not a problem that you would have with something like cells, right? So a fundamental question in stress research and in neuroscience research as a whole is, where along this sort of complexity spectrum do you want to run your experiments, right? How do you balance complexity, or some would say realism, versus the ability to really precisely control every aspect of the organism you're trying to study? Well, tonight we're going to hear from three awesome neuroscientists who have each decided to answer that question in a different way. First up will be Dr. Michael Hart, who will be talking about his work in C. elegans, i.e. worms. And he'll be answering the question that I'm sure some of you are already asking, which is, how in the world can we learn anything useful about stress using worms? After that, we'll hear from Dr. Deborah Bangasser, who uses rodent models, so mice and rats, to study how males and females react to stress differently. And then finally, we'll hear from Dr. Alison Mackey, who will tell us about how we can use techniques like MRI, which is what's sort of up on the slide here, to study stress and stress responses in real living humans. So, we'll sort of move along this scale here as we go through the talks tonight. And in each talk, we'll get to hear about some of the unique advantages and disadvantages of studying each of these, types of organisms.